Single crystal X-ray diffractometry is the oldest and most precise form of X-ray crystallography, where X-ray crystallography is the famous technique that was used against Rosalind Franklin's consent in Watson and Crick's quest to determine the structure of DNA. In the process known as X-ray diffraction, X-rays strike an object, in our case this crystalline solid, and the X-rays are diffracted by the small spaces between particles. The size of the spaces dictate the magnitude of diffraction, which in turn dictates the final position on the photographic plate. X-ray diffractometers can be partitioned into three main components: the X-ray tube, the sample holder, and the X-ray detector. The very first step of X-ray diffraction begins with the creation of X-rays. The first component of the X-ray diffractometer is the X-ray tube. Say we have a cathode ray tube that produces X-rays. Cathode ray tubes work on the principle of heating a filament to produce a beam of electrons. These electrons are accelerated to a target by the use of an applied voltage. It is when the electrons have the adequate quanta of energy to dislodge inner electrons of the target material, which is usually molybdenum, that a spectrum of X-rays is produced. Diffraction itself requires a specific wavelength, so the X-rays are either passed through a foil or a crystal monochromator. After the X-rays have passed through the wavelength selector, the rays are collimated. The X-rays strike the sample and constructive interference occurs when the Bragg's equation is satisfied. It is the diffracted X-rays that are recorded by the detector, so the resultant diffracted direction of the lattice are obtained by changing the angle of incidence along with the orientation of the centered sample. Furthermore, the ability to change angles to obtain the full diffraction readout is done through the use of the goniometer. Seen here is the derivative for Bragg's law. For anything to be read by the detector, X-rays must reach the detector, so constructive interference of X-rays must occur. And constructive interference occurs only when Bragg's law is satisfied. The technique gathers detailed information about crystal structure, which includes bond angles, bond lengths, site ordering information, and unit cell dimensions. Here are the related advantages and disadvantages of the technique. It is important to note that it is only with the use of specialized chambers that one can determine the high temperature and pressure phases. The Swafford paper is an example of how X-ray diffractometers are used in research-based application. The nature of their research was to determine the precise structure, size, and composition of homogeneously alloyed cadmium, sulfur X, selenium 1-X nanocrystals. The samples were synthesized by the research in a single prolytic method, which was based on work done by the researchers Yu and Pang. One conclusion the researchers reached was the sulfur-rich seeds found in the nanocrystals do not affect the size and composition of nanocrystals. This means that all nanocrystals larger than the sulfur seeds are considered homogeneous, which is supported by their X-ray diffraction spectra. And lastly, here are my sources.